Okay, in today's video, we're going to talk about the digestion and absorption of food types. We're going to look at the accessory organs of the digestive system now. And then we're also going to talk about how it is controlled, neural and endocrine regulation. As we said, we're going to talk about our accessory organs, and here we have the liver, and this is only showing you part of it, but really they kind of look like these hexagonal shapes like this, and they have a central vein in the middle where blood is going to dump into. On the corner, or at the junction of each of these, there are three vessels, and I'm trying to draw those with our three, and that's these right here. And these are called the portal triad. There is one portal vein bringing blood in, bringing deoxygenated blood in. There is one hepatic artery bringing oxygenated blood in. And then there is one bile duct coming out, so bringing bile out. So if we look at this a little closer, here we have our portal triad with our three vessels. And our portal vein, and I'll draw this in blue to denote deoxygenated blood. Our portal vein is bringing deoxygenated blood from our digestive system. So from our small intestines, from our mesentery, deoxygenated blood is coming in here. And the purpose of this is not to oxygenate the tissue of the liver like normal, right? If blood were coming to an organ. The purpose of this is to cleanse the blood, detoxify the blood. Maybe you've heard about how the liver is a detoxifier. It is going to detoxify blood here. And so what that means is that anything you eat, is going to go into the blood, and that those nutrients are absorbed in our small intestine. But before that blood goes into our general circulation via the inferior vena cava, it's going to go to the liver first via these portal veins. And it's going to go into the liver first to make sure that it's okay. And it's going to take out any waste products. And so we're going to talk about that today. So this is bringing in deoxygenated blood. It does have a lot of nutrients in it though, because it is coming from our small intestine where we just ate a bunch of food and molecules are coming in through there. And so these nutrients can feed into the liver, but this is where blood is detoxified here. So oxygenated blood is going to come in, of course, because we need to oxygenate our liver. And it's going to come in via the hepatic artery. And hopefully you remember that from anatomy. And what's interesting here is that we find that these two blood types, these two bloods, I guess, mix. And they end up mixing, and they are traveling through these sinusoids, where just these like ducts through our liver lobules, which are those hexagonal shapes. That's a lobule. And then they're going to exit via the central vein, which is going to feed into a hepatic vein, which is going to feed into our inferior vena cava. Blood here is deoxygenated, so really this should be blue, not purple, because it is oxygenating these tissues at the same time. At the same time that all this is happening, these cells here of the liver lobules are called hepatocytes, and they are going to detoxify our blood. We'll talk about the mechanisms that happens. Another thing that they do though is that they secrete bile. And so I'm drawing in green here because bile is actually green. And notice that the bile is completely separate from blood. and it's going through these vessels called bile canaliculi. If you can remember from anatomy, canaliculi just means like little vessels, little canals. And so these are going to carry bile out via the bile ducts. So a big takeaway here is that although there is a lot of blood flow and a lot of bile flow, they do not mix together. So the liver is going to take in blood, as we see here, via our hepatic portal vein, and those are going to feed into our lobules, and they're going to get detoxified. But some of those molecules that came in via the hepatic portal vein, they could actually get recirculated and go out via the bile. And so it may come back out via the bile and come back out the common bile duct into the small intestine. And then because it's going back to where, the blood got circulated in the first place, it actually may get circulated back via the hepatic portal vein. And so it can actually recirculate. And we find that some compounds do that, like certain drugs, for example. And this is called enterohepatic circulation. So some examples of this, drugs, for example, ampicillin, penicillin, these may be recirculated because they get brought into the liver, but then they get released into the bile, and so they're going to go back out into the small intestine and then reintroduced into the liver, and so it goes through via enterohepatic circulation. Some naturally occurring compounds do this, and these are called endogenous compounds. For example, bile salts, urobilinogen, and lecithin. Those are naturally occurring compounds that are a result of breakdown of other compounds that can go into this enterohepatic circulation. So our liver, we've been talking about the functionality a little bit, but it is going to detoxify blood, and the way that it does that in a big way is via bile, as we talked about in that enterohepatic circulation. Bile is going to help remove waste, and that waste could be toxic, it could be drugs, which means outside chemicals. It can be hormone. It's just going to break down certain things, and it could use bile to do that. Another way is by these immune system cells called Kupffer cells, and these are going to do phagocytosis, and so they're going to break down like bacteria, for example. Another way that liver can detoxify blood is by taking molecules that are pretty toxic to us, like ammonia, and making them into less toxic waste compounds, like urea. And another factor of liver that can do this is something called cytochrome P450, which can assist in breakdown of drugs. So the detoxification of blood is through these multiple, 4 different mechanisms that it can do. So bile is a big part of that, but this production of bile by the hepatocytes is important for this function of liver. Something that's also really cool about liver is it is really good at regenerating. It's really good at regeneration. So you can cut off like a third of your liver and it will regenerate and regrow back because it can kind of adjust into these progenitor mitotic cells that are going to regenerate. And then once a full liver has regenerated, it'll stop. And so it does mitosis really well when it needs to. However, you may have heard of like damage of the liver, damage of those hepatocytes when you think about like alcohol or cirrhosis of the liver for alcoholics. That would be unreversible damage of liver tissue, and so it doesn't have that regenerative capacity. The liver can remove or add glucose to the blood, and so if you think about terms of fasting, for example, in terms of fasting, liver glycogen, so glycogen that's stored in the liver, can be broken down into glucose. Amino acids can be broken down into glucose. Fatty acids can be broken down into ketone bodies, and maybe you've heard of the keto diet. This is the mechanism with which they're trying to recreate or do. And then in terms of having like a big meal, like eating, the liver can take out glucose from the blood to produce glycogen or to produce fats, like produce triglycerides. It can also produce plasma proteins such as albumin, clotting factors, cholesterol is a big one that we'll talk about, and globulins. In the liver, we also have metabolism of heme, and from that bilirubin, and then metabolism of bilirubin too. So our red blood cells, which have hemoglobin, are going to be broken down into heme and going to release this iron, which will be recycled. The pigment here is going to produce a molecule called bilirubin in the liver. This will then produce a conjugated bilirubin from the liver, which is hydrophobic. And so this will piggyback on proteins to move around in the blood. Bilirubin can be eliminated by mixing with bile, which will be then eliminated through the intestines into the feces. So this image here is showing the hepatic portal system with blood going in via the hepatic portal vein, and there can be some molecules that are going to enter the enterohepatic circulation where they can get released with bile so that they go back into the small intestine, and they can even go back into the circulation. Bilirubin can be one of those things. It can also be excreted. Bilirubin, with that conjugated bilirubin, bacteria can transfer that into urobilinogen, and this is actually pigmented as well. This is actually what gives feces the brown color that it is, urobilinogen. And then if this bilirubin or urobilinogen continues on in this hepatic portal system and maybe even reaches through the general circulation, what can happen is it can go into the kidney where it will be excreted in the kidney, and urobilinogen is excreted in the kidney, and that's actually what gives the pigment, the amber pigment of urine as well. So the way that bile works is, say you have a big globule of fat or oil. What it does is it emulsifies fats. And the way that it does this is it takes large globules of fat and they make them into smaller globules of fat by mixing them with these bile acids. And these bile acids are amphipathic, where they have a hydrophilic head and a hydrophobic tail. And so what happens is these micelles form around these flat globules. And when it is able to break them down into smaller fat globules, as we can see here, this increases the surface area. So when it comes into contact with enzymes such as lipase, there is more surface area for the enzyme to come into contact with these to break them down. So jaundice, a clinical relative example here is jaundice. Jaundice is caused by the yellowing of tissues due to increased bilirubin. And again, remember we said that bilirubin is pigment. It's the pigment of our red blood cells, which is also what allows for this yellowing of the tissue. And this can be caused by a few things. Gallstones. Gallstones are where the bile excretion, so maybe a bile duct is blocked, and so then you get an accumulation of bile salts, and this could be really painful. This can cause increased bilirubin. Another thing would be erythroblastosis fetalis, which can occur if mother and baby have different blood types, specifically RH, rhesus blood type. This can cause a big high rate of red blood cell destruction. If you have a high rate of red blood cell destruction, you're going to have high bilirubin. Physiological jaundice at birth. These babies don't have the liver enzymes that conjugate bilirubin in the first place, so it can't be eliminated, and so they can't they can't release bilirubin, and so there is buildup within the liver. And the treatment to this would be to expose infants to blue light so that bilirubin can then be eliminated in the bile or urine. So here we have the gallbladder, and we will talk about how when there is no more digestion occurring, like food has moved past, this duodenal papilla kind of closes up and it causes backflow of bile, and then that backflow of bile goes into the cystic duct and gets stored in the gallbladder. That's how it goes there, even though, it is produced in the liver up here. So the gallbladder is going to store bile. It also can concentrate bile. And as we said before, if there is a blockage of a bile duct, this can form a gallstone. Also, excess cholesterol can form gallstones too. And then we have the pancreas, which I'll show you an image of in just a second. But we have two portions of it. The endocrine portion that's going to secrete hormone, and then the exocrine portion that is going to release secretions. And those secretions are going to be digestive enzymes as well as bicarbonate. So here is our pancreas. And as you can see here, I will color this in red. We have a pancreatic duct here, and those exocrine secretions are going to come out via these pancreatic ducts. And as you can see, the acinar cells, which are going to produce those digestive enzymes, are going to release their secretions out in the pancreatic secretions. But then we also have different cells, and these cells are called pancreatic islets or islets of Langerhans. These are going to have endocrine function, and they are going to produce insulin and glucagon to control blood glucose. So here it is showing close up. And again, we have our acinar cells, which are exocrine, producing our enzymes or bicarbonate. And those are going to release their secretions into the pancreatic duct. And here in this histology picture, they are the darker color. And then the lighter colored cells are those pancreatic islets or islets of Langerhans. And these are going to be our endocrine portion that are going to produce insulin and glucagon. So one of those enzymes, which is shown here, that is released by the pancreas, is called trypsinogen. Similar to pepsinogen, this is inactive, and similar to pepsinogen, it also digests protein. It digests protein. So again, we want it to be inactive because we wouldn't want it to digest the cell that it comes from. It can come into contact when it comes into contact with a surface epithelial cell of the small intestine of the duodenum. These are showing enterokinase cells. That is going to activate it, and then it becomes trypsin, which is the active protein digesting enzyme. Trypsin can also activate other inactive enzymes that come from the pancreatic juice to make them active. And so inactive enzymes are called zymogens. And again, especially in terms of digestion, we wouldn't want our enzymes to be active in their active state within the cell. So this is a little bit of review from the first week that we had this class, where we talked about breakdown of molecules, and these are showing hydrolysis events where water is used to break down these biomolecules. We have carbohydrates, we have proteins, and we have lipids. And as you can see, they're being broken down into their smallest constituents. Monosaccharides for carbohydrates, amino acids for our proteins, and then fatty acids and glycerol. So these are the monomers of these polymer biomolecules. So let's talk about digestion a little bit. And digestion, well, we're talking specifically about chemical digestion here, because we're looking at the enzymes, because mechanical digestion has to do with like teeth chomping, mastication, as well as like the muscle action of the GI tract and the stomach. But when we talk about chemical digestion, we're talking about the enzymes here. And right away, it begins in the mouth with salivary amylase to digest carbohydrates. This is going to break down starch, and I believe it's in a future slide. In the duodenum, the pancreas also has an amylase, which again will also break down starch. And then in the duodenum, as well as in the rest of the small intestine, we have enzymes called brush border enzymes, which are in the microvilli of our villi. And those are going to further break down our biomolecules into their absolute smallest constituents so that they can be absorbed through the intestinal epithelium. Glucose moves through the intestinal epithelium and it's secreted into our blood capillaries, which is what insulin and glucagon are controlling. This begins in the stomach with pepsin. Remember, pepsin has to be activated by HCl. The inactive form is pepsinogen. In the duodenum, we have other protein digesting enzymes. Remember, we said trypsin, we have chymotrypsin, and then two others, elastase and carboxypeptidase. And again, these are all for digestion of protein. We have an enzyme at the brush border that's going to complete digestion called aminopeptidase to break down our peptide bonds into single amino acids, which then can move through the epithelium within the duodenum and into the blood. So here we have a polypeptide chain, a protein chain, And this is showing some peptidases that are within the lumen of the small intestine. We have brush border enzymes that are going to further break it down, which allow for amino acids to come through our epithelium into our blood. And now for the digestion and absorption of lipids, where things are a little different. In the duodenum, thanks to the liver and gallbladder, we have our lipids emulsified by bile salts. And so what that means is, again, they're going to take large globules of fats, lipids, and break them down into smaller globules so that these lipases that they come into contact with have a greater surface area and they're able to more easily digest these lipids. So one enzyme, and this is where it begins, is in the duodenum, is the pancreatic lipase. So one of those digestive enzymes from the pancreas is lipase to break these triglycerides into free fatty acids and glycerol. What's different here, though, is that these large fat droplets are going to be emulsified into smaller fat droplets. They're going to be digested by pancreatic lipase to form these fatty acids and monoglycerides. And then they're going to produce these micelles. And remember, micelles are just a mixture of hydrophilic and hydrophobic components, where the hydrophilic heads are sticking out so that they can travel through our aqueous blood plasma. From here, these are going to attach onto a protein and produce something called a chylomicron. A chylomicron allows for larger fats, larger lipids to travel with the addition of a protein. And these are larger, and so they can't actually travel through blood, and they do not travel through blood. And as you can see here, it's traveling through lymph fluid via these vessels called lacteals. And these lacteals are found in the villi of the small intestine. Lipoproteins carry lipids in blood too. So chylomicrons are going to carry lipids via lacteals. But if lipids are small enough, lipoproteins can carry lipids in the blood. There are two types here: low density lipoproteins and high density lipoproteins. High density lipoproteins are kind of considered good cholesterol because they just go to the liver and they are degraded. Low density lipoproteins are considered kind of bad because they can move through tissues to be stored, and they can actually cause buildup within arterial walls. They can cause buildup in arterial walls. And so this can cause plaques down the line. And so these are not very good for you. All right, the last part of our digestive physiology is talking about how it is regulated. And we can break that up into two types: extrinsic, meaning outside of the GI tract. And here we have control via the autonomic nervous system. And this is to do with the parasympathetic and sympathetic divisions. The parasympathetic is going to increase GI function, so that's going to increase secretion, increase movement, increase motility, and it can travel via cranial nerve 10 vagus nerve. The sympathetic division is going to have opposing effects, antagonistic effects from that. Something else that can feed into this autonomic control or through the vagus nerve is via the enteric nervous system. And I know enteric is written down below, but hear me out, which is made up of the myenteric and submucosal plexuses. So what actually happens is the sensory information from the myenteric and submucosal plexus can actually feed into the vagus nerve as well. And so this is called extrinsic afferent information. Extrinsic afferent information. So it is extrinsic because it's coming via the vagus nerve, but it's kind of like in the middle because it's coming from the enteric nervous system. And then we have intrinsic control, which again, here we see the enteric nervous system. And again, that includes the myenteric and submucosal plexuses. So let's talk about these a little bit. The myenteric plexus is like another way to think of the enteric nervous system is kind of like the brain of the gut. It's kind of like the nervous system nervous tissue of the gut. And there are two plexuses that control here. The myenteric plexus, and we're going to find this in the muscularis externa layer. So the circular layer as well as the longitudinal layer. And this is going to control motility. So think about peristalsis, right? The movement or segmentation here. This can also control the interstitial cells of Cajal because those are going to also control those muscle movements. And then we have the submucosal layer, the submucosal plexus, which is found in the submucosal layer. And so this is going to control glandular secretions, right? Because that is part of our submucosal layer. And so those are two separate plexuses that make up our enteric nervous system, which is really part of the GI tract. So it is considered intrinsic control. So these are going to include sensory neurons, interneurons, and because they do not go to the brain, because it is considered brain of the gut, these have neural reflexes about them. And this is how peristalsis and segmentation with these interstitial cells of Cajal are controlled. We have hormones of the GI tract, and we're just going to focus on these three major ones. One secreted by the stomach, by G cells, are going to stimulate the release of hydrochloric acid by parietal cells. It can stimulate the secretion of pepsinogen to digest protein by chief cells in its inactive form. And it also allows for maintenance of the mucosa layer of our stomach. So that's the job of the hormone gastrin. The small intestine can secrete a hormone called secretin, which is going to stimulate water secretion or bicarbonate secretion in pancreatic juice. And we didn't talk about this, but the pancreas also releases bicarbonate, and that is going to allow for neutralizing our HCl. And then it is going to impact the actions of CCK on the pancreas. So there is another final hormone here called CCK, and that can stimulate contraction of our gallbladder to release bile. It can stimulate more secretion of our pancreatic juice enzymes. It helps in maintaining the structure of our acinar cells. But lastly, it can also inhibit motility of our stomach and secretion. And this may occur or want to occur at the end of digestion, in the intestinal phase of digestion. So there are three phases of gastric function. First off, before you even taste or begin digesting, there is the sight of food, the smell of food, and that causes the vagus nerve to already stimulate secretions. And the taste of food will include that too. The food is going to be put into the mouth and brought into the stomach, which will bring about the gastric phase, and that is going to allow for stretching of stomach cells as well as those activation of secretions. So we talked about chief cells, secreting pepsinogen. We talked about parietal cells secreting HCl. We talked about G cells secreting gastrin. What else did we talk about? We talked about ECL cells, which are going to be in response to this. And so all of these secretions are occurring because of the arrival of food in the stomach. As soon as food moves into the intestines, now we are at the intestinal phase. And this is going to stop stomach activity by that action of CCK, which allows for digestion of chyme. So this is just showing kind of a lot of what we just spoke about. The G cells here are going to secrete that hormone gastrin. Well, let me start from the beginning. When amino acids come into the stomach, that is going to encourage G cells to secrete gastrin. The vagus nerve can also stimulate G cells to secrete gastrin, which is going to encourage your ECL cells to secrete histamine, and that will cause your parietal cells to secrete hydrochloric acid. And then this excess of hydrochloric acid is going to reduce the pH of the stomach, the gastric juices, which is then going to inhibit or reduce the secretions of the G cells. So that'll stop it. And so this is a negative feedback loop.